Silk gland of Eri Silkworm Samia, cynthia ricini. Histological analysis: silkworms secrete silk from the specialized organ called the silk gland. In lepidopteran insects, one pair of silk glands originate in the labial segment and extend throughout the abdomen. The ducts of each gland merge at the anterior extremity to form a common duct that opens at the base of a hollow spine, the spinneret. The spinneret is positioned at the tip of the mouthpart hypopharynx. The epithelia of the silk and salivary glands are ectodermal in origin and are lined with cuticles composed of mainly the polysaccharide chitin and a blend of proteins. The silk gland of the Eri Silkworm Samia, cynthia ricini is a pair of modified tubular structures folded in a zigzag pattern. Each tube extends from the labial to the posterior abdominal segment. The weight of the whole silk gland is around 0.60 grams, the length is around 1 meter, and the diameter of the posterior silk gland is around 604 micrometers. They are suspended in the hemocoel and bathed with hemolymph. It shows a high degree of bilateral symmetry with each tube positioned on either side of the digestive tract. The proximal ends of the silk gland tube on both sides connect to form a common duct before opening through the hypopharyngeal spinneret. In the posterior abdominal segment, the distal ends terminate blindly near the ventral integument. Based on the size and histological structure, the silk gland can be divided into 3 regions: the anterior, the middle, and the posterior silk gland. The silk gland tubes of Samia, cynthia ricini taper from the posterior portion towards the anterior, without distinct boundary of anterior, middle, and posterior silk gland. However, in the mulberry silkworm Bombyx mori, the middle silk gland is larger in diameter compared to the anterior and posterior silk glands. Histological structure of the silk gland of Samia, cynthia ricini. The histological structure changes gradually from the proximal to the distal end, which differentiates the silk gland into anterior, middle, and posterior regions with indistinct boundaries. The columnar epithelial cells are arranged in a single layer, surrounding the large central lumen structures of the anterior silk gland. The cells of the anterior region are non-secretory. These cells have distinct nuclei. A prominent tunica intima forms the boundary of the lumen. Thin tunica propria forms the external membrane of the epithelial cells, structures of the middle silk gland. The nuclei of the columnar epithelial cells of the middle silk gland appear irregular in shape. The transverse sections of middle silk gland of 5th instar day 1 wandering C. ricini larvae are seen with less prominent tunica intima. Less distinct tunica propria makes the outer layer of epithelial cells. Protein secretions by the epithelial cells of this region look like continuous flow threads flowing towards the central protein mass secreted from the posterior region. The secretions of middle silk gland form a distinct layer around central protein mass. Histological structures of the posterior silk gland. The transverse sections of the posterior silk gland of 5th instar day 1 wandering Samia, cynthia ricini larvae are seen with a large lumen. The nuclei are convoluted. Secreted protein globules are visible in the luminal border detaching from the indistinct tunica intima. These globules coalesce with each other in the lumen and condense towards the center of the lumen, forming a fibroin column. Tunica propria forms the outer layer of the silk gland epithelial cell.